The aim of this procedure is to acquire time-lapse fluorescence images of growing E. coli colonies expressing CoTrAC constructs to identify the number of newly produced protein molecules. This is accomplished by first preparing a sample of cells co-expressing a protein of interest fused to a fluorescent CoTrAC reporter, and a protease that specifically cleaves between the CoTrAC reporter and the protein of interest. The second step is to prepare a gel of low melting temperature agarose made with M9 minimal media. Next, an agarose gel pad is prepared for use with a temperature-controlled sample chamber. The E. coli cells are placed on the gel and the chamber is assembled. The final step is to place the chamber on a fluorescence microscope and image several growing cell colonies at regular intervals over multiple generations, photobleaching newly produced CoTrAC reporter molecules at each interval. Ultimately, CoTrAC is used to acquire time traces of protein production in live cells with single molecule and single cell resolution.
The main advantage of this technique over existing single-cell methods using fluorescence protein fusion is that the production of any kinds of protein molecules can be followed with single-molecule precision and without affecting that molecule's functions.
To engineer the strain, insert sequences encoding a membrane localization.